Hello everyone. Welcome to the new session of Anatomy of Flowering Plants Part 2. So in this session we are going to discuss about the dicot stem and monocot stem. We have already discussed about the dicot roots and monocot root, and in this session we are going to study about the stem part. Okay, so let us start this session. Anatomy of dicotyledonous and monocotyledonous plant. So first of all, we will discuss about the dicotyledonous stem. So to study about the dicotyledonous stem, we need a transverse section. We have to see the transverse section of any typical young dicotyledonous stem. So first of all, what you do, you will take a dicotyledonous stem and you will cut the transverse section of it. Now, how to cut the transverse section? You will take a pith that can be made up of potato tuber, and you will, in between that, you will cut a slice in between the tuber, and you will place that a young dicotyledonous stem. And when you cut the thin section of this pith, that is the potato, the same time you will cut the very fine transverse section of the stem also, right, with the help of blade. Now, the transverse section of a typical young dicotyledonous stem shows the epidermis in the outermost protective layer of the stem. So I have already told you that if you see the any organ of the dicotyledonous or a monocotyledonous plant, what you see, the outer surface. The outer surface is made up of the epidermal tissue. Okay, there is an epidermal tissue system. We have already talked about it. So epidermal tissue is made up of the epidermis layer. So here also, in the transverse section, you see the outer surface is made up of the epidermal cells. Okay. So here you can see the dicot and monocot stem. This is the diagrammatic as well as the cellular structure. So if you see the diagrammatic structure, you can see the epidermis layer, and outer epidermis layer is having some epidermal hair also, right? So in the cellular structure, you can see the epidermis and it is having some epidermal hairs, right? Now below this layer, epidermis layer, there is a collenchyma cells. There is a layers of the collenchyma cells. Okay, 4-5 layers of the collenchyma cells. And below this collenchyma, there is a parenchyma cells. Okay, and in between the parenchyma, you all know that there are having the intercellular spaces in between. Okay, the intercellular spaces in between, and that is the parenchyma. Now below this, you can see the endodermis. Okay, so the outer collenchyma, the parenchyma, endodermis, this all make the ground tissue. Okay, or you can say the cortex. Now below this endodermis, there is a pericycle layer. Okay, so pericycle layer is there. It is maybe some cells thick, and after that you can see the presence of the vascular bundle. So here you can see in the case of dicot stem, the vascular bundles are arranged in a ring form. All the vascular bundles are in a ring-like arrangement, right? And this is showing, the cellular structure is showing one of the vascular bundle. So if you see one of the vascular bundle, how it is, the phloem is present outside, right? And the xylem is present inside. The metaxylem and the protoxylem arrangement is there. Metaxylem is toward the inner side and the protoxylem is toward the outer side, right? In between the phloem and xylem cells, you can see the presence of the cambium layer. So it is a conjoined and open type of vascular bundle. It is an open type of vascular bundle, cambium, right? Present in between the phloem and xylem cells. Now, if you see the monocot stem, the monocot is having again the epidermis, and below the epidermis there is an hypodermis. Okay, and the vascular bundles are scattered. Vascular bundles are scattered all over the stem region. It is not showing any arrangement of the ring-like structure. Okay, it is scattered all over the stem. So here you can see the dicot and monocot stem. Okay. Now, anatomy of the dicotyledonous and monocotyledonous stem, and that is if you see this epidermis then covered with a thin layer of cuticle, the epidermis having a thin layer of cuticle, and it may bear the trichomes and the few stomata. So sometimes they are having some stomata present on the outer stem region, and they are having the hair-like structure that is called as the trichomes. Those hair-like structure which is present on the stem, they are called as the trichomes. The cells arranged in a multiple layers between the epidermis and pericycle constitute the cortex. Okay, and this many cells, you can see the collenchyma cells are there, you can see the parenchyma, then endodermis, then pericycle. So all these up to the pericycle, it constitute the cortex region, right? The cells arrange in multiple layers between the epidermis and pericycle. The arrangement of cells between the epidermis and pericycle, it is called as the cortex. Okay, now it consists three subzones. Now we have to see what are these three subzones. The outer is hypodermis. Okay, hypodermis, which is made up of few layers of collenchymatous cells. Okay, we have already discussed about this. The hypodermis, it is made up of the collenchyma cells. Okay, and which is having no intercellular spaces in between. And it is present just below the epidermis, which provide mechanical strength to the young stem. So their presence provide the mechanical strength to the young stem. So this collenchyma is there because it is also having a lot of flexibility, right? So you know that the plants with the help of wind, they can move this direction or to other direction, and this is just because of this collenchyma. They are not getting broken down, right? And they are having the flexibility due to the presence of this collenchymatous. So they are providing the mechanical strength to the young stem. Okay, now the cortical layers. Next comes to the cortical layers. So below epidermis consists of rounded thin, this hypodermis, there consists of the thin walled round structure cells, which is called as parenchymatous cells, and with conspicuous intercellular spaces. There are lot many intercellular spaces present, right? So they are the parenchymatous cells present just below the hypodermis. Okay, the innermost layer of the cortex is called the endodermis. So it is started from the collenchyma, that is the hypodermis, this cortex region. It is started just below the epidermis. And what is present just below the epidermis? Hypodermis cells, which is made up of the collenchyma, right? And the innermost layer of the cortex, if you talk about the innermost layer of the cortex, then it is the endodermis, means the layer which is present outside the pericycle, endodermis. So started from the collenchymatous cell, that is the hypodermis, and up to the endodermis, it constitute the cortex region. It constitute the cortex region. The cells of the endodermis are rich in starch grains. Very, very important. This endodermis is having a lot of starch grain, and the layer is also referred to as the starch sheath due to the presence of this. Okay, due to the presence of this lot of starch grain, because this whatever the food material is synthesized in the plant, it is converted into the starch, and these starch is stored in the case of this endodermis. Okay, so sometimes it is referred to as the starch sheath. Pericycle is present on the inner side of the endodermis. So where this pericycle is present? Pericycle is present the inner side of the endodermis. Okay, just below the endodermis, towards the inside, it is having the pericycle layer, and above the phloem in the form of semi-lunar patches of the sclerenchyma. So this pericycle, it is made up of the sclerenchyma, and it is very, very important, right? It is present above the phloem cells, and you can see the semi-lunar patches, mean it is made up of 4 to 5 layers of the cells, and divide and redivide and give out the lateral roots. So lateral in the case of root, if they are present, so they gives the pericycle give rise to the lateral root. But here we are talking about the stem. So in the stem, they are not giving rise the any type of root like lateral root, but yes, it divide and redivide to form the cork. Okay, the outer cork layer, it is formed from this pericycle only. Okay. Now, in between the vascular bundle, there are few layers of radially placed parenchymatous cells which constitute the medullary rays. Very, very important point. Now, in between the vascular bundle, if you see the vascular bundle, there are so many cells, okay, so many cells that is present radially. They are present radially in between the vascular bundle, and these are the parenchymatous cells. These are made up of the parenchymatous cells which constitute the medullary rays. So those parenchymatous cells present in between the vascular bundle, they are forming the medullary rays. A large number of vascular bundles are arranged in a ring. So this is a very unique feature of the dicotyledonous system, that vascular bundles are arranged in a form of ring-like structure. Very unique. Ring-like structure. All are present in the ring-like structure, and this ring arrangement of the vascular bundle is a characteristic of the dicot stem. So this type of structure you see in the case of dicot stem only, right? So this ring-like arrangement of the vascular bundle is a characteristic of the dicot stem. Now, if you talk about the vascular bundle, what type of vascular bundles they are having? Each vascular bundle is conjoined. If you see the vascular bundle, then they are conjoined. Open means they are having the cambium layer with enlarged protoxylem. Their protoxylem is enlarged type, right? So the vascular bundles are conjoined. The xylem and phloem are present at the same radius, and in between the xylem and phloem there is a presence of cambium. So this type of vascular bundle is called as a open type of vascular bundles, right? So this is a very unique feature of the dicotyledonous stem. We will see in the case of monocotyledonous, there is a huge difference between the dicot and monocot on the basis of this vascular bundle only, right? The large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. So there is a big pith present in the case of the dicotyledonous stem. So large number of rounded, what type of cells are present in the case of pith? Parenchymatous cells. This, if you see the shape of the cell, then they are rounded and they are having lots and lots of intercellular spaces because they are oval or the round in shape. So they forms the intercellular spaces in between, which occupy the central portion of the stem constitute the pith, and they are present in the central portion of the stem, which is forming the pith region. So this is in the case of dicot stem that you see the such type of pith structure. Now we will talk about the monocotyledonous stem. The monocotyledonous stem is very much different. Okay, they are very different from the dicotyledonous stem. So we have all the three different type of tissue system. They are present in the case of dicot stem. That is the epidermal tissue system. Then there is a ground tissue system. Then there is a vascular tissue system, right? So all the three different types of systems are present in the case of monocot stem also, but there is a lot of difference in the case of all the three systems. So let us see what type of anatomical structures the monocotyledonous stems are having. The monocot stem has a sclerenchymatous hypodermis. Very, very important. Sclerenchymatous. Okay. So below the epidermis layer, there is a sclerenchymatous layer. What we have seen in the case of dicot stem is having collenchymatous type of hypodermis. Collenchymatous type of hypodermis. But here, what you see is sclerenchymatous type of hypodermis. A large numbers of scattered vascular bundle, each surrounded by the sclerenchymatous bundle sheath and large conspicuous parenchymatous ground tissue. Now you have seen the difference between the hypodermis of the monocot and the dicot. Now we will see what is the arrangement of the vascular bundle. In the case of monocot, the vascular bundles are scattered. Okay, it is scattered all over the stem. It is scattered all over the stem, and they are surrounded by the sclerenchymatous bundle sheath. They are surrounded by the sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Okay, the parenchymatous type of ground tissue they are having. But what is absent here? What is absent here? There are no cortex. There is no endodermis. There is no pericycle. You see in the case of monocotyledonous stem. So this is the showing the huge difference between the dicotyledonous stems and the monocotyledonous stem, right? So we have seen the difference on the basis of the epidermis, because the epidermis, they are not giving rise any type of stem or the hair-like structure, the trichomes. Okay. So in the case of monocotyledonous, we see the difference between the epidermis layer. We see the difference between the hypodermis layer. In the case of dicot, they are having the collenchymatous cells. Here, the sclerenchymatous cells. No cortex region is present in the case of monocotyledonous stems. And there is a very, you can say, the demarcated, conspicuous cortex region is found in the case of dicot stem. Vascular bundles are also arrangement is very, very different. Dicot, they are having the ring arrangement is in the form of ring-like structure. All the vascular bundles are arranged in a ring-like structure. But here in the case of monocot stem, the vascular bundles are scattered, okay, and they are surrounded by the sclerenchymatous bundle sheath. They are surrounded by the sclerenchymatous bundle sheath. They are large conspicuous parenchymatous ground tissue. If you see the ground tissue, then there are made up of the parenchymatous cells. So here you can see in the case of monocot stem, they are having the epidermis, right? But epidermis is not having any hair-like structure. In the case of dicot stem, you can see they are having the epidermal layer. So we can see the difference on the basis of epidermis, epidermal tissue. Okay, now coming to the hypodermis. Hypodermis here, it is made up of sclerenchymatous tissue. Whereas in the case of dicot, you can see the collenchymatous tissue. Cortex region is there, no cortical region is there. The vascular bundles are scattered all over. Smaller vascular bundles are present at the periphery and larger vascular bundles are in the center region. Okay. In the case of dicot, you can see the vascular bundles are arranged in a ring-like structure. Pith, you can see the large pith in the case of dicot stem, whereas smaller pith, the pith is very, very small in the case of monocot stem. Vascular bundles are conjoined and closed. If you see one single vascular bundle in the case of monocotyledonous stem, then the vascular bundles are conjoined, means xylem and phloems are compacted together in a form of vascular bundle, but they are closed type. Closed type means no presence of cambium. Cambium is totally absent in the case of monocotyledonous stem. Okay, peripheral vascular bundles are generally smaller than the centrally located one. Okay, peripheral vascular bundles are smaller. They are small inside, and the centrally located vascular bundles are larger. The phloem parenchyma is absent. Okay, phloem parenchyma is absent in the case of monocot. So this on the presence of this component of the phloem, there we see the difference on this basis also. Parenchyma, phloem parenchyma is absent, and the water containing cavities are present within the vascular bundles. Okay. Okay, now let us see what is the difference between the leaf structure. Means the dicot leaf and monocot leaf. So here we have taken the dorsiventral, means the dicotyledonous leaf, because the dicotyledonous leaf, there we see the two surfaces, the dorsal surface and the ventral surface, and that is why they are called as the dorsiventral leaf. The vertical section of a dorsiventral leaf through the lamina shows the three main parts. So again, you have to cut the vertical section of the dorsiventral leaf, dicot leaf, and from where you cut the lamina part, the main blade that is having the tissue, or that is called as the lamina. You take vertical cut section of the leaf, and what you see under the microscope, you see the three main parts. Again, here comes the epidermis. I've told you that epidermis is the outer covering of all the parts of the plant, right? Whether it is a root, whether it is a stem and the leaf. So epidermis is present on the surface of the leaf also. Now, below the epidermis, what you see? Mesophyll cells. You see the mesophyll cells and the vascular system. So three different things you see in the case of internal or the anatomy of the leaf, internal structure of the leaf or the anatomy of the leaf. So you are having the epidermis system, you are having the mesophyll, and you are having the vascular tissue system, right? So let us see in detail what is the arrangement of the tissue in the dicotyledonous leaf. So epidermis, which covered both the upper surface, adaxial epidermis, and the lower surface, abaxial epidermis of the leaf, has a conspicuous cuticle. So if you see the epidermis, then it is covered the both. Means epidermis is present at the upper surface also and the lower surface also. Okay. Upper surface, which is also called as the adaxial epidermis. Upper surface is also called as the adaxial epidermis, and lower surface is called as the abaxial epidermis. Okay. Now they are having the conspicuous cuticle. The cuticle layer is present at the surface of the upper as well as the lower epidermis. But yes, at the upper epidermis, it is more confined because to minimize the rate of transpiration. And you see the stomata are mostly confined to the lower surface. Okay. In the case of dicot, sometimes it is totally absent. But if some plant is having the stomata on both the surfaces, then lower surface is having the more number of stomata if you compare it with the upper surface, right? So let us see the presence of stomata. The abaxial epidermis generally bears more stomata than the adaxial epidermis, and you all know that this stomata is the modification of the epidermal tissue only. So it is the modification of the epidermis. So the later may even lack stomata. Sometimes the lower epidermis is not having any stomata. Okay. So the tissue between the upper and the lower epidermis is called the mesophyll tissue. Okay. So if you see the tissue which is present between the upper epidermis and the lower epidermis, it is called as the mesophyll tissue. Okay. Mesophyll, which possesses the chloroplast. If you see the mesophyll, then it is having a lot of chloroplast and carry out the process of photosynthesis, and it is made up of the parenchyma. Okay, so mesophyll cells, they are having chloroplast and they are carrying out the process of photosynthesis, and it is made up of the parenchyma cells. It has two types of cells, the palisade parenchyma and the spongy parenchyma. So two types of parenchyma cells are there. Palisade, which this palisade parenchyma are made up of elongated cells. The cells of the palisade parenchyma are elongated in shape, which are arranged vertically and parallel to each other. All the cells are of the palisade parenchyma are arranged elongated, okay, and they are vertically placed, and no intercellular spaces is present in between the palisade parenchyma. But what about the spongy parenchyma? Spongy parenchyma are oval and rounded, and loosely arranged spongy parenchyma is situated between the palisade cells and extend to the lower epidermis. So mostly this spongy parenchyma cells are present at the lower epidermis. There are numerous large spaces and air cavities present in between the cells. So the spongy parenchyma cells are having lots and lots of air cavities in between these cells. Okay, vascular system include the vascular bundle, which can be seen in the veins and the midrib. So when you cut the cross section, okay, so this cross section is crosses between from the midrib region and the veins also. That is showing the, when you see the internal structure, so that part is showing the presence of vascular bundle. So this vascular bundle for forming the vascular tissue system, right? And it is showing in the veins and the midrib region. The size of the vascular bundles depend upon the size of the veins. Okay, if the veins of the secondary order, tertiary order, so it will the small, okay, small vascular bundle. But if it is of the primary order or the main midrib, then it will have the larger vascular bundle. The veins vary in thickness in the reticulate venation of the dicot leaf. So we have the different layers of, you can say, the different order of the veins, right? So if the veins are thick, they are having the reticulate venation. So the dicot leaves will show the such type of veins, and the vascular bundle surrounded by the thick layer of bundle sheath cells. So here you can see a thick layer of bundle sheath cells is surrounding the vascular bundles in the case of dicot. So we can see the figure that is showing the xylem in the vascular bundle. So this is the dorsiventral structure of the dicot leaf. So here you can see the upper epidermis, here you can see the lower epidermis. Lower epidermis is showing the presence of the stomata, and you can see the two types of mesophyll cells, the palisade mesophyll cells, which is showing the presence of the chloroplast, okay, and the in between the palisade cells you cannot see any like intercellular spaces, right? And lower region, it is having the spongy mesophyll cells, okay, and in between they are having the large air cavities, right? So some stomatal cavities are also there, and it is having the stomata, so the gaseous exchange takes place here. If you see the vascular bundles, it is having the bundle sheath cells surrounding, and the vascular tissues are present in this region. Now just look at the monocot. The monocot is having the upper epidermis and the lower epidermis, and the upper epidermis is also having the stomata, and the lower epidermis is also having the stomata. So upper and lower epidermis is having the equal number of stomata in the case of monocot stem. So this is the huge difference, right? And the cuticle layer is also present. But what you see, the major difference here in the case of monocot, the palisade and spongy parenchyma type of cells are mesophyll cells are not there, right? They are having just a mesophyll cells. And the air cavities, what about the air cavities? No air cavities are there, only the substomatal cavity is there. So this is showing the huge difference, right? And see the arrangement of this vascular bundles also. Vascular bundles is having the large, it is not having any bundle sheath cells. Okay, so let us see. Isobilateral monocotyledonous leaf is also called as the isobilateral type of leaf. The anatomy of the isobilateral leaf is similar to the dorsiventral leaf in many ways. So they are having all the things, but there are some modifications. It shows the following characteristics differences. What are those? In an isobilateral leaf, the stomata are present on both the surface of the epidermis. With this, we have already discussed. The stomata are confined on the both the surface, and the mesophyll cell is not differentiated into palisade and the parenchyma type. So we have seen this structure. In grasses, the certain adaxial epidermis cells elongate along the veins, modify themselves into the large empty colorless cells, right? These are called as the bulbiform cells. Bulbiform cells, okay, in grasses. So adaxial epidermis, the veins, along the veins, they modify themselves and they form the empty colorless cells. These are called as the bulbiform cells. When the bulliform cells, okay, cells in the leaves have the absorbed water and are turgid, the leaf surface is exposed. So they absorb the water, they become turgid, and the leaf surface become exposed. So for the exposure to the leaf surface, these bulliform cells are present in the monocot stem. When they are flaccid due to the water stress, they make the leaves curl inward and minimize the water loss. So because the stomata are present on the upper and lower surface equally. So how to minimize the rate of transpiration when there is a water stress? So these bulliform cells becomes flaccid, they curl the leaves inward, and in this way they can minimize the water loss. The parallel venation in the monocot leaf is reticulated in the near smaller size of the vascular bundle, except in the main veins, as seen in the vertical sections of the leaf. So this is all about this session. Okay. Thank you very much for attending this session. We will meet again. Till then, take care and bye.